Assalamualaikum and a very good morning to my first panel, Technologist Dr. Nur Faizah Muhammad Razali, and to my second panel, Dr. Muhammad Haikal Abdul Aziz. Before I proceed with my presentation, I would like to introduce you my supervisor, Prof. Madya Dr. Anzasari Muhammad Qasim, and my co-supervisor, Dr. Mariam Firdaus Binti Nordin. Today, I would like to present my project proposal, which the title is The Effect of Different Wall Materials, Inulin, Gum Arabic, and Maltodextrin in Spray Drying Process for Curcuma Subspecies Microencapsulation Efficiency. So here my list of contents consists introduction, literature review, methodology, and expected result. Introduction. The first part of introduction is background research. There are 128 species of curcuma plants. Medicinal plants chosen to be studied in this research are Curcuma longa and Curcuma xanthorrhiza. Both species have bioactive compounds that are beneficial to human health. For example, the bioactive compounds can treat Alzheimer's, inflammation disease, and cancer due to exhibition on biological activities. The extracted bioactive compounds will be new development in food and medical field, but there are some challenges in developing these bioactive compounds. Bioactive compounds are low solubility, low stability, and low bioavailability. For the second part is problem statement. So, why do I do this study? Actually, there are many benefits of bioactive compounds from this plant to human health. Many countries such as Indonesia, Malaysia, and Thailand have applied this plant as traditional medicine. In this era, modern medicine will be easier and more precise in determining the diagnosis. However, there are some problems in bringing up the bioactive compounds into food and medical field. So, how can we overcome the problem? First, propose the appropriate method for bioactive compounds, which is microencapsulation. In microencapsulation, spray drying technique is the particular process. Actually, there are many techniques of microencapsulation such as freeze drying and centrifugal extrusion. The method has been chosen because microencapsulation is the technology that encapsulates bioactive compounds. So, to make it more effective microencapsulated product, using different wall materials such as inulin, gum arabic, and maltodextrin will be applied in this process. The third part is research objective. So, there are three objectives in this research, which the first one is to microencapsulate the curcuma subspecies extract with different wall materials. The second one is to analyze the physicochemical characteristic of microencapsulated extract. And the last is to evaluate the microencapsulation efficiency of each microencapsulated curcuma subspecies. The last part in introduction is scopes. Okay, so there are three scopes in this research, similar as objective. So the first one is microencapsulating process of curcuma subspecies by spray drying method from curcuma extraction using different wall materials such as Arabic gum, maltodextrin, and inulin. The next one is producing microencapsulated extracts of curcuma subspecies in order to analyze physicochemical characteristics in terms of physical, chemical, and structural. And the last one is evaluation of microencapsulation efficiency of curcuma subspecies by testing sample on Curcuma longa and Curcuma xanthorrhiza. Literature review. Okay, so in the first part of literature review, I show the table of different and similarity of bioactive compounds in both curcuma, which are Curcuma longa and Curcuma xanthorrhiza. So here we can see Similarity between Curcuma longa and Curcuma xanthorrhiza is both species have three bioactive compounds, which is a curcumin, dimethoxycurcumin, and bis-dimethoxycurcumin. And where the difference of both species is Curcuma longa consists of germacran, while Curcuma xanthorrhiza consists of xanthorrhizol as bioactive compounds. Details on literature review is the extraction process will be done in subcritical water extraction. Among other extraction process, the SWE is more green as it uses the water to extract the substance. Water is non-toxic and can be used to extract many types of plants. As we know, the bioactive compounds will be encapsulated by spray drying. Based on spray drying parameters such as temperature, it is a suitable technique to encapsulate bioactive compounds. From previous research that I found, it also has few justification why spray drying is more better than other techniques like freeze drying and centrifugal extrusion. So there are three wall materials such as inulin, gum arabic, and maltodextrin used in this process. In this case, the formulation between those materials can give better result in improving the solubility and stability of the microencapsulated product. Okay, so here I attach the table that show the product, the final product of moisture content that consists from the formulation of wall material. The next part is methodology. In this part, there is a flowchart of microencapsulation of Curcuma subspecies. Okay, so the first part in this process of microencapsulation is sample preparation of Curcuma longa and Curcuma xanthorrhiza. And the second part is extraction of both Curcuma subspecies using subcritical water extraction, which is SWE, and followed by preparation of wall materials, which is inulin, gum arabic, and maltodextrin, and then microencapsulated by spray drying. And the last is characterization and analysis of subcritical water extraction, extracted bioactive compounds. So we have three analysis, which is physical characterization, structural characterization, and functional characterization. Details on microencapsulation of Curcuma subspecies by spray drying. So the rhizomes of Curcuma longa and Curcuma xanthorrhiza will be prepared. Dry both Curcuma subspecies in order to obtain the powder form of both species. Extract the powder of Curcuma subspecies into subcritical water extraction. Obtain the extracted product from SWE of bioactive compound from Curcuma subspecies, and then undergo microencapsulating by spray dryer using different wall materials such as I mentioned before, like inulin, gum arabic, and maltodextrin. And the last one, we evaluate the characteristic of each microencapsulated product from both Curcuma subspecies. Expected result. Okay, so for the last part is expected result, which is the first one is for moisture content. Formulation of modified starch and inulin will be shown the great value of moisture content among others wall materials. The formulation of these wall materials will produce the most dry microencapsulated product. So the second result from analysis is solubility. For solubility, the best wall material is gum arabic. The properties of gum arabic itself will increase the solubility of both Curcuma species. And the third one is morphology. The formulation between modified starch and maltodextrin is process higher preparation spherical particles. The structure produced by the formulation is showing the better product for microencapsulation. And the last analysis is total flavonoid content. Among other 128 curcuma subspecies, Curcuma longa and Curcuma xanthorrhiza will give the highest total of flavonoid compounds. So Curcuma longa give 741.36, and while Curcuma xanthorrhiza gives 220.53. So here I attach the Gantt chart that I categorize by task and timeline, which is 13 weeks. Thank you.